Hello, everyone. I hope you're all energized and ready because today we're going to explore one of the most fascinating and essential topics in immunology, the lymphoid organs. Now, whether you're preparing for NEET or you're pursuing your bachelor's in biology, this topic forms a cornerstone of understanding how our immune system works. Let me ask you something. Have you ever wondered how your body knows how to fight off a cold? Or why your lymph nodes swell when you're sick? Or even how vaccines work to keep us safe? The answers to all of these questions start right here, with lymphoid organs. Think of the immune system as a country's defense force. Now, every effective military needs several things: a place to train new recruits, storage for weapons, strategic planning centers, and frontline stations for quick response. Similarly, our immune system has its own set of structures, the lymphoid organs, that help it prepare for and react to threats. These organs are where immune cells are created, matured, stored, and activated. Today, we're going to classify these lymphoid organs into two groups: primary and secondary. Primary lymphoid organs are like training academies. This is where immune cells, especially lymphocytes, originate and learn how to distinguish between what belongs to our body and what doesn't. The two major players here are the bone marrow and the thymus. Secondary lymphoid organs, in contrast, are like battlefields where these trained lymphocytes encounter antigens and get activated. These include lymph nodes, spleen, and MALT, mucosa-associated lymphoid tissue. We'll look at each of these in detail, not just anatomically but functionally. This includes histological views, immune cell movement, and clinical significance. By the end, you'll understand how your immune system is orchestrated across different tissues to provide constant protection. Before we begin exploring each organ, it's important to be clear about our goals today. These objectives guide not only what you learn, but also how you'll apply it in exams or further studies. Understand what lymphoid organs are and why they matter. At the most basic level, you'll know what structures are included under this term and how they support the immune system. You'll understand why immune cells don't just float around randomly in the body. Differentiate between primary and secondary lymphoid organs. This is a key distinction. Primary organs are responsible for lymphocyte production and maturation, while secondary organs are involved in lymphocyte activation and immune response. Remember, structure follows function. Identify and describe the bone marrow and thymus. You'll learn where B cells and T cells mature, how they develop self-tolerance, and what happens if they don't. We'll talk about positive and negative selection in the thymus and how this ensures autoimmunity doesn't occur. Understand the structure and role of secondary lymphoid organs, such as lymph nodes, the spleen, and MALT. You'll explore how these organs serve as checkpoints and meeting points for antigens and immune cells. You'll also learn about germinal centers, antigen presentation, and memory cell formation. Apply the knowledge clinically. Whether it's understanding why lymph nodes swell during infection, or how the thymus is involved in diseases like DiGeorge syndrome, we'll make sure this knowledge connects to real-world biology. Exam readiness. By the end, you'll be able to handle NEET-style MCQs and short-answer questions that often trip students up, like identifying which organ lacks afferlymphatics, or understanding why splenectomy increases infection risk. Before diving into lymphoid organs, let's review the immune system itself, because lymphoid organs don't work in isolation. They are part of a beautifully coordinated system. The immune system protects the body from pathogens, tumors, and abnormal cells. It is divided into two main arms. Innate immunity. This is your body's first responder. It's nonspecific, rapid, and includes physical barriers like skin, chemical barriers like stomach acid, and cellular defenses like macrophages and neutrophils. Adaptive immunity. This is specific, targeted, and has memory. It involves lymphocytes, B and T cells, that recognize specific antigens and respond more effectively upon second exposure. Adaptive immunity is slower to develop, but much more powerful and precise. Now, here's the key point. Lymphocytes, B and T cells, don't spontaneously become functional. They require maturation, a process that happens in primary lymphoid organs. Once mature, They travel to secondary lymphoid organs, where they encounter antigens and initiate immune responses. This entire immune choreography is dependent on proper function and structure of lymphoid tissues. You'll see how B cells undergo clonal selection and activation in lymph nodes. You'll understand how T cells recognize antigen MHC complexes thanks to their training in the thymus. The link between organ structure and immune function is the central theme we'll carry throughout this lecture. Let's now get into the meat of our topic, the classification of lymphoid organs. All lymphoid organs fall into two categories, primary and secondary. Why this division? Because it reflects their function in the immune life cycle. Primary lymphoid organs are where lymphocytes are born and trained. These are bone marrow, where B cells mature and all blood cells are produced. Thymus, where T cells undergo rigorous education and selection. These organs are essential for immunocompetence, the ability of immune cells to distinguish self from non-self. Without proper function here, the immune system would either be inactive or attack the body itself. Secondary lymphoid organs are where immune responses are initiated. These include lymph nodes, stationed along lymphatic vessels. They filter lymph and present antigens. Spleen filters blood, traps blood-borne antigens, and manages immune responses in the bloodstream. MALT, a diverse group of tissues guarding mucosal surfaces like the respiratory tract, GI tract, etc. One key difference: primary lymphoid organs do not directly interact with antigens. That role belongs to secondary organs. This classification isn't just academic. It forms the framework for understanding immune surveillance, disease defense, and vaccine design. Let's talk about one of the most foundational sites in our immune system, the bone marrow. This organ is central not just to immunity but to life itself, because it's where all our blood cells, red, white, and platelets, are born. This process is called hematopoiesis. Bone marrow is found inside bones. In children, it's present in the long bones like the femur, but in adults, it's mostly located in flat bones like the sternum, vertebrae, ribs, and pelvis. Now there are two types of marrow. Red marrow. This is active in hematopoiesis. It's rich in stem cells and found in spongy bone areas. Yellow marrow. This is mostly made of fat and serves as energy storage. It can convert to red marrow in emergencies, like severe blood loss. Let's zoom in on immunity. Bone marrow is where B lymphocytes, the B cells, develop and mature. The B in B cell actually refers to the bursa of Fabricius in birds, but in humans, their entire development happens here in the marrow. They go through gene rearrangement to form unique receptors, basically customizing their weapons, and then they are tested for self-reactivity. If a B cell reacts too strongly to our own body, it's eliminated in a process called central tolerance. This prevents autoimmunity. Apart from B cells, the bone marrow also produces precursors for T cells. These cells leave the bone marrow and travel to the thymus to continue their maturation. Clinically, bone marrow is incredibly significant. In conditions like leukemia, the marrow gets overtaken by cancerous white cells. In aplastic anemia, the marrow fails to produce enough blood cells. In both cases, a bone marrow transplant (BMT) can be life-saving. Healthy donor marrow can restore a patient's blood cell production and immune function. So in short, bone marrow is not just a tissue inside your bones. It's the origin of your immune system's army. Without it, there would be no B cells, no T cell precursors, no defense. Now that we've explored the bone marrow, let's move to the second primary lymphoid organ, the thymus. This is the specialized school for T lymphocytes or T cells, and it's absolutely vital to our adaptive immune system. First off, where is it? The thymus is a bilobed organ located just above the heart, behind the sternum. In young individuals, particularly children, it's relatively large and very active. But here's something fascinating. As we age, the thymus gradually shrinks and gets replaced by fatty tissue. This process is called involution. That's why the thymus is much more active during childhood. It's when the immune system is being trained and programmed for life. Functionally, the thymus is like a military academy for T cells. Immature T cells, which are produced in the bone marrow, migrate to the thymus to undergo maturation and selection. Now, this maturation is not just about growth, it's about education. The thymus teaches these cells how to distinguish between the body's own cells and foreign invaders. The thymus is divided into two main zones, the cortex and the medulla. In the cortex, immature T cells begin their training. This is where positive selection takes place. Only those cells that can recognize the body's MHC, major histocompatibility complex, molecules survive. The rest are eliminated. It's a bit brutal, but essential. Then the surviving cells move to the medulla, where negative selection happens. Here, any T cell that reacts too strongly to self-antigens is eliminated.
Why?
Because we don't want our immune system attacking our own tissues. That would lead to autoimmune diseases. So, only the T cells that are both functional and self-tolerant are allowed to leave the thymus and enter the bloodstream. Let me pause and emphasize something important for NEET and your degree exams. The thymus is the only organ where T cell selection occurs. It doesn't fight infections directly. It prepares the soldiers. And here's a clinical connection. In DeGeorge syndrome, a child is born without a functional thymus. As a result, they have very poor T cell immunity and are highly vulnerable to infections. Also, thymic tumors or hyperplasia can be associated with autoimmune conditions like myasthenia gravis, something we explore more deeply at the degree level. So remember, the thymus isn't just an anatomical structure. It's a quality control checkpoint for your immune army. And like any good academy, it ensures that only the best and most disciplined cells are sent out to protect the body. Alright, now we move from the academies, the primary lymphoid organs, to the battlefront of the immune system, the lymph nodes. Lymph nodes are small, bean-shaped structures scattered throughout the body, particularly in areas like the neck, armpits, groin, and abdomen. They are connected by a vast network of lymphatic vessels, like highways, that carry a clear fluid called lymph. This lymph fluid contains antigens, immune cells, waste products, and sometimes even pathogens. Now, why are lymph nodes so important? Think of them as immune surveillance checkpoints. Whenever lymph passes through them, the immune system inspects it thoroughly. If there's an invader, say a virus or bacteria, the lymph node responds rapidly, activating and expanding immune cells. That's why lymph nodes swell during infections. They're filled with rapidly dividing lymphocytes and macrophages working overtime. Anatomically, each lymph node has three main regions. The cortex, which contains B-cell-rich follicles. This is where B cells are activated and can form germinal centers if there's an infection. The paracortex, where T cells hang out and interact with dendritic cells presenting antigens. The medulla, which contains plasma cells, the antibody factories, and macrophages. Lymph enters the node via afferent lymphatic vessels, percolates through the cortex and medulla, and exits through efferent vessels at the hilum. One common NEET trick question: which lymphoid organ has both afferent and efferent lymphatics? Answer: lymph nodes, unlike the spleen, which only has efferent lymphatics. Functionally, lymph nodes are where adaptive immune responses are initiated. Dendritic cells bring antigens here and present them to naive T and B cells. If the cells recognize the antigen, they get activated, divide, and some become memory cells, while others fight the current threat. From a clinical perspective, swollen lymph nodes are a key diagnostic sign. They're often enlarged in infections, autoimmune diseases, and cancers, particularly lymphomas or metastases from other tumors. Here's something else to remember. Sentinel lymph nodes are the first nodes to receive lymph drainage from a cancer site, like in breast cancer. Biopsying these can help assess metastasis. In essence, lymph nodes are highly dynamic organs. They're not passive filters. They're like strategic military outposts, constantly watching, learning, and responding to biological threats. Now let's talk about the spleen, a secondary lymphoid organ that often doesn't get as much attention, but trust me, it's a powerhouse when it comes to both immunity and blood filtration. The spleen is located in the upper left quadrant of your abdomen, just beneath the diaphragm and close to the stomach. Unlike lymph nodes, which filter lymph, the The spleen is the only lymphoid organ that filters blood directly. So think of it as a security checkpoint for your bloodstream, scanning every drop for pathogens, damaged cells, or foreign particles. Structurally, the spleen is divided into two distinct zones, the red pulp and the white pulp. Let's start with the red pulp. This region is primarily responsible for filtering and recycling old or damaged red blood cells, RBCs. As blood flows slowly through this area, macrophages engulf and break down aged RBCs, salvaging useful components like iron and heme. It's like a biological recycling center. The red pulp also stores platelets and acts as a reservoir during times of massive blood loss. Now the white pulp, this is the immune function center. It's rich in lymphocytes, especially T and B cells. It surrounds a central arteriole, forming something called the periarteriolar lymphoid sheath, PALS, where T cells dominate. Nearby follicles are where B cells reside. So, when pathogens enter the bloodstream, they can be detected and neutralized right here. In short, the white pulp initiates immune responses against blood-borne antigens. So, how does this play out in real life? Say a bacterium enters your bloodstream, maybe through a deep cut or an infected wound. The spleen acts fast, filtering the blood and activating lymphocytes to mount an immune response. Without it, your body would be much slower to react to systemic infections. That brings us to a very important clinical angle. What happens if the spleen is removed? This is called a splenectomy, which might be done after trauma, for example, in a car accident, or in diseases like hereditary spherocytosis. But without a spleen, the body becomes vulnerable to certain types of bacterial infections, especially encapsulated bacteria like Streptococcus pneumoniae, Haemophilus influenza, and Neisseria meningitidis. These organisms are typically cleared efficiently by the spleen's macrophages. That's why splenic individuals are advised to get vaccines against these pathogens and sometimes require long-term antibiotics. One more NEET relevant point. Unlike lymph nodes, the spleen does not have afferent lymphatics. It receives antigens through the blood, not through the lymphatic system. So in summary, the spleen is both a filter and a fighter. It clears old RBCs, detects blood-borne infections, and helps activate immune responses. It's the perfect blend of a recycling plant and a rapid response unit. Up next, we'll zoom into the third major secondary lymphoid tissue, MALT, or mucosa-associated lymphoid tissue, which protects all the entry points of your body. So far, we've looked at bone marrow and thymus, where lymphocytes mature, and we've looked at lymph nodes and the spleen, where immune responses are activated in tissue and blood, respectively. But what about the places where pathogens usually enter the body, like the nose, mouth, gut, and lungs? That's where MALT comes in. MALT is a diffuse system of lymphoid tissue located in the mucosal linings of the body, the moist surfaces that are constantly exposed to the external environment. This includes the respiratory tract, gastrointestinal tract, urogenital tract, and more. It acts as a first line of immune defense in these vulnerable areas. Let's break this down. You've probably heard of tonsils, right? These are part of MALT, and they guard the entry of air and food through the mouth and nose. Then there's Peyer's patches, which are found in the walls of the small intestine. They're basically surveillance posts in the digestive tract. You also have appendix, bronchus-associated lymphoid tissue, BALT, and nasopharynx-associated lymphoid tissue, NELT, all subdivisions of MALT. Now, what do all these structures do? They detect antigens from pathogens entering through mucosal surfaces. Specialized cells called M cells in the epithelium help sample antigens and deliver them to underlying lymphoid tissue, where T and B cells can mount a response. Here's a key point. MALT is rich in IgA-producing plasma cells. Immunoglobulin A, IgA, is the primary antibody found in mucosal secretions, like saliva, tears, mucus, and breast milk. IgA helps neutralize pathogens right at the point of entry before they can even cause an infection. What's especially fascinating is that MALT is highly dynamic and adaptive. For example, when you're exposed to a pathogen in the gut, it triggers an immune response in the local Peyer's patches, but it can also lead to the generation of memory cells that patrol other mucosal areas. This crosstalk makes MALT a system-wide mucosal defense network. From a clinical standpoint, inflammation or abnormal immune activity in MALT can lead to conditions like inflammatory bowel disease, IBD, or MALT lymphomas, especially in the stomach, often linked with Helicobacter pylori infection. This shows how local immunity can go awry if the balance is disrupted. One NEET relevant fact: MALT accounts for nearly 50% of all lymphoid tissue in the body. That's huge. It reminds us how important mucosal immunity is, because, after all, most pathogens don't barge in through the skin. They sneak in through what we breathe, eat, or touch. So, MALT might not have the grandeur of the spleen or the order of a lymph node, but it is quite possibly the most heavily used part of your immune system. Now that we've covered the major lymphoid organs individually, it's time to bring it all together in a clear, comparative way. This slide shows a summary table that highlights the key features of each lymphoid organ, primary and secondary, side by side. Why is this important? Because many questions in NEET and your biology exams will expect you to quickly identify differences. For example, which organ is the site of T-cell maturation? Which one filters blood? Which organ involutes with age? And so on. Let's walk through the table. First, bone marrow. It's the site of hematopoiesis, meaning all blood cells, including lymphocytes, are generated here. B-cell maturation happens here too. Structurally, it contains both red marrow, active in blood cell formation, and yellow marrow, mostly fat. Clinically, we think of bone marrow transplants, which are life-saving in leukemia and other blood disorders. Then, the thymus. This is where T-cell maturation and selection occur. It has a unique bilobe structure with cortex and medulla zones. It's large and active in children but undergoes age-related involution in adults. Disorders here can cause immunodeficiency. Moving to lymph nodes, they are the immune checkpoints along lymphatic vessels. Key features include the cortex, paracortex, and medulla regions. They receive lymph via afferent lymphatics and send it out through efferent lymphatics. This organ is crucial in antigen presentation and lymphocyte activation. The spleen is unique because it filters blood directly rather than lymph. It has red pulp for RBC destruction and white pulp rich in lymphocytes for immune surveillance. It has only efferent lymphatics. Removal of the spleen raises risks of bacterial infections. Lastly, MALT. This is a diffuse system located in mucosal linings like tonsils and Peyer's patches. It specializes in mucosal immunity and produces IgA antibodies. It's essential in defending against pathogens at entry points. Now that we understand the anatomy and function of lymphoid organs, it's crucial to connect this knowledge to clinical scenarios you might encounter, both in exams and in real life. Let's start with severe combined immunodeficiency, or SCID. This is a genetic disorder that affects both T and B lymphocytes, essentially wiping out the adaptive immune response. The root of the problem often lies in the primary lymphoid organs, especially the thymus and bone marrow, where lymphocyte development goes awry. Babies born with SCID are extremely vulnerable to infections. Without a functioning thymus to mature T cells and bone marrow to produce B cells, their immune system is like an army with no soldiers. Treatment often involves bone marrow transplantation to restore immune function, highlighting the vital role of these primary lymphoid organs. Next, let's talk about AIDS, acquired immunodeficiency syndrome caused by HIV infection. Here, the virus primarily targets CD4+ T cells, which mature in the thymus and reside in secondary lymphoid organs like lymph nodes and the spleen. As AIDS progresses, you get secondary lymphoid atrophy, meaning these organs shrink or lose cellularity. Lymph nodes become depleted of their immune cells, weakening the body's ability to fight infections. This explains why AIDS patients are susceptible to opportunistic infections. Another important clinical point relates to cancer metastasis. Lymph nodes play a dual role. They're part of the immune defense, but also often the first site where cancer cells spread from primary tumors. For example, in breast cancer or melanoma, tumor cells migrate through lymphatic vessels and lodge in regional lymph nodes, causing lymphadenopathy, enlarged lymph nodes. This is why sentinel lymph node biopsy is important in cancer staging. It helps determine the extent of spread and guides treatment. All right, as we come to the end of today's lecture, let's quickly recap the main points so everything stays fresh in your mind. First, remember that the immune system relies on specialized organs called lymphoid organs, which are broadly divided into primary and secondary. The primary lymphoid organs, bone marrow and thymus, are where lymphocytes are born and trained. Bone marrow is the site of hematopoiesis, the formation of all blood cells, and specifically B-cell maturation. The thymus is the exclusive site of T-cell maturation and selection, where positive and negative selection ensure only the right T-cells enter the bloodstream. Then we move on to the secondary lymphoid organs, the front lines where immune cells gather and mount responses. These include lymph nodes, which filter lymph and are key sites for antigen presentation and lymphocyte activation. The spleen, unique in filtering blood, also removes old red blood cells and provides immune surveillance for blood-borne pathogens. And then there's MALT, the diffuse lymphoid tissue that protects mucosal surfaces, our body's entry points for most pathogens. Clinically, these organs are critical. Disorders like SCID affect primary organs, leading to severe immunodeficiency. AIDS causes destruction of T cells and atrophy of secondary lymphoid organs, compromising immunity. The spread of cancer often involves lymph nodes, making them key in diagnosis and staging. And removal of the spleen puts patients at risk for serious infections. To sum up, lymphoid organs are essential hubs for creating, educating, and deploying immune cells. Their proper function keeps us healthy and protected from the multitude of pathogens we encounter daily. Thank you all for your attention. Feel free to ask any questions or clarify doubts. I'm here to help you master this crucial topic. Also, don't forget to subscribe to the channel for more lessons.